Okay, hey, we are beginning a brand new, we're beginning a brand new unit today. That unit is mitosis, is what we're dealing with. All right, and in mitosis, we're going to be talking about cell division. All right, it's kind of since we're nearing the end of cells. You know, cells are the basic unit of structure and function in living things, so they're always going to be kind of in the background. Of what we're talking about. But this is our last thing we're going to be specifically talking about cell parts. We will a little bit with DNA next unit as well, but we're nearing the end. Thankfully, I can only do so much for cells here. So anyway, mitosis. Today we're going to talk briefly about the stages of mitosis. You've probably already had this in some way, shape, or form. So not going to spend a ton of time talking about the different phases, but what's important to know is the order of the phases and what's going on with the chromosomes in those phases. All right. If we think back, for human beings, the number of chromosomes that we have, think about it right now. All right. Hopefully you're talking about 46 chromosomes, and they come in 23 pairs. All right. So 46 chromosomes in 23 pairs. The important thing about that is that number is consistent. Okay, it's not like it's like an instruction manual. Just adding a bunch of pages to the instruction manual doesn't have any benefit. Taking pages out of the instruction manual certainly doesn't have any benefit. And so for human beings, 46 is that sweet spot. And we need to make sure that every single cell has those 46 chromosomes. So most of our discussions about mitosis is going to be talking about how do we maintain that number of chromosomes. And we start off with interphase. Now, interphase is kind of a cheat, all right, because interphase is actually the time when the cell is not dividing. Okay, so a lot of people won't even include this in mitosis because this is just your cell doing cell things. This is your stomach cell being a stomach cell, a blood cell being a blood cell, a bone cell being a bone cell, those kind of things. So this is the time between mitosis, so between cell divisions. However, at the very end of interphase, when it's about ready to go into mitosis, we are going to copy those chromosomes. All right, so we want to make sure that we make a copy of all the chromosomes getting ready to split. So step one, interphase. Step two is prophase. All right, prophase. Chromosomes pair up. That's how you remember that. The two P's there. Chromosomes pair up in prophase. So we see our sketch in the middle there. Chromosomes have clearly paired up into homologous pairs. There's a cell part that we maybe briefly talked about, but I left out for on purpose here. And these are centrioles, represented by kind of the little tubular looking things that are brown on the diagram below there. Centrioles, their main job is to aid in cell division. All right, so these two things polarize, meaning they go to the opposite ends of the cell, and they're going to be a big driver for what's going to pull the cell apart there. Okay, so the chromosomes have paired up, and the centrioles have polarized in prophase. Step number two. Stage three, metaphase. Metaphase is going to be the easiest one to identify because in metaphase, cells are lined up in the middle. Sorry, chromosomes are lined up in the middle of the cell. The nucleus is gone. All right, it has dissolved. And what's left is we have these chromosomes lined up in the middle of the cell, paired up. And then the spindle fibers are actually forming and they're starting to attach to those chromosomes. So if we look at a sketch of it here they're lined up in the middle. And then those spindle fibers are these blue things. They're not really that straight in real life, but they hook on to those chromosome pairs. So metaphase, things are lined up in the middle, two M's there for stage number three. Number four, anaphase. Chromosomes are being pulled apart. So anaphase apart. That is my memory trick for that one. And it's stage number four. And so those spindle fibers start to shorten, and our chromosome pairs that are rolling here are going to start to pull to the two sides of the cell. All right, so the spindle fibers are pulling the chromosomes apart. You can see a sketch of it in the lower left-hand corner as well. And last phase-ish is telophase. All right, look at the diagram below. In telophase, we now have two cells that each have 46 chromosomes. I have not clearly drawn 46 chromosomes, just 4, but in human cells, each cell will have 46 chromosomes. And you can see below the nucleus, those dotted lines of nuclei are starting to form, and that two cells are going to pinch in the middle to separate at the end there. So we have two new daughter cells, each with 46 chromosomes in telophase. No great memory trick for telophase other than telo and two new cells in there. And then last and definitely least, cytokinesis. A lot of times this gets lumped in with telophase because the other organelles duplicate as well. All right, that's something that I kind of just gloss over. Is like, oh, we're so concerned with the chromosomes. Okay, cytokinesis, they're gonna, cells are gonna split up the endoplasmic reticulum, the mitochondria, the ribosomes, that kind of thing as well. It's just not as vital as making sure that we have all the right number of chromosomes. So cytokinesis, cell organelles duplicate in two new cells, and there's a clear division, and we went from one cell to two cells. So those are the stages of mitosis in order, with a brief description of what's going on. And remember, this is how organisms grow, right? That's how you go from a one-cell being to a trillion-cell being, is by mitosis over and over and over again.